defining protective metabolic pathways in triple-negative breast cancer. So based on Canadian breast cancer statistics, we know that 1 in 8 women will be diagnosed throughout their lifetime, and this equated to about 29,000 women in 2023. What's more concerning is that despite advancements in early diagnosis and treatments, 1 in 36 women will still die due to metastatic events, and this translated to about 5,400 Canadian deaths last year. Clinically, breast tumors are molecularly classified into 5 subtypes, and this can be seen here on the slide. These align with the expression of histopathological biomarkers used in breast cancer diagnosis. So this includes 2 hormone receptors, the estrogen and progesterone receptor, as well as human epidermal growth factor receptor 2. The expression or lack thereof of these receptors can inform clinical information for both clinicians and patients. And this includes information such as treatment options and prognosis. So we can see here at the bottom that triple-negative breast cancer is the most aggressive subtype, and this is associated with the worst prognosis. As its name suggests, TNBC is characterized by lack of estrogen, progesterone, and HER2 expression. And this is about 12 to 17% of breast cancers. Due to its aggressive phenotype, these patients have a high risk of metastasis. So over 1/3 of patients will develop metastasis throughout the course of their disease. The lack of targetable receptor expression and high degree of metastases make this very difficult to treat. So as I previously mentioned, receptor status can inform treatment options. And because there is a lack of receptors, there's also a lack of targetable drug options. So treatment options consist of a more systemic approach, and this includes things like surgical resection, radiation, systemic cytotoxic chemotherapeutic agents, and immunotherapy. And by nature, because it's a systemic approach, these are really harmful and more toxic to the body. Patients do tend to respond well to therapies initially. However, these patients do often develop resistance, which leads to low overall survival and a high risk of recurrence. So this is a lot of challenges. And because of this, it's evident that there's more work that needs to be done to better understand which patients are most susceptible to metastases and identify potential therapeutic targets to improve clinical outcomes. Previous research has focused a lot on studying the genomic and transcriptomic landscapes of TNBC. So studies have separated TNBC tumors into 4 transcriptomic subtypes, and these each have distinct characteristics. There are associated therapies that have been developed for these subtypes, but they've had limited success. So systemic chemotherapies do remain the standard of treatment for these patients. Due to the high risk of recurrence associated with TNBC and the limited success of these treatments, it's evident that further research is needed. So current studies in our lab, amongst others, are focused on exploring the TNBC metabolome as a way to get a more integrated understanding because the metabolome is impacted upstream by the genome, transcriptome, and the proteome. The metabolome involves small molecule signaling, and this can be investigated to profile cellular metabolites and examine how they affect the phenotype and function of cells. So this can allow us to identify the presence of distinct metabolic signals in TNBC with the hopes of identifying tumors that are more likely to be aggressive and gain a better understanding of the TNBC landscape as a whole to improve clinical outcomes. So what is metabolism? This is essentially a series of chemical reactions and changes in the body that give you the materials that you need to grow, reproduce, and stay healthy. So essentially what's happening is you eat food, it gets broken down into things like amino acids, glucose, fatty acids, and then these get used to build proteins, generate energy, and synthesize lipids, and this all contributes to the normal physiological function in the human body. These metabolic processes are highly regulated so that you can maintain homeostasis. And so the way that we think about like whole body metabolism is similar to how we think about it in a cell. Cells need fuel to function, and the things that they need depend on their particular function. So because cells need fuel to function, they make use of the nutrients that are available to them. But cancer cells tend to be a little bit more greedy. They like to soak up all the fuel, all the nutrients in their environment to account for their needs. And this allows them to rapidly proliferate and survive in the tumor microenvironment. When they've taken what they want, or they're no longer happy in their environment, they then move to a new location, and then they exhaust the resources in that new location. So that homeostasis piece that I was talking about before is essentially lost. A common example that many of you may know is the Warburg effect. So cells take up the glucose at a higher rate and favor the production of lactate, and this throws off the balance of sugar metabolism. This dysregulated pathway is less efficient in terms of ATP generation, but it is a lot faster and it still generates the nucleic acids that the cancer cells require. There's also a lack of fatty acid regulation. So both the synthesis and oxidation pathways of fatty acids are active concurrently. So bringing it back to why we're here, studying the metabolome allows us to look at what is fueling these highly aggressive and metastatic cells and look at how we can possibly shut down or reroute these food stores. So interestingly, there was a study conducted by Gong et al., and they identified 3 metabolic pathway-based subtypes, MPS 1 through 3, and they each have their own distinct metabolic features, and they have distinct prognoses, metabolic distributions, and genomic alterations. So the first one on the top here in blue is the lipogenic subtype, characterized by upregulated lipid metabolism. Moving down to MPS 2 in red, we have the glycolytic subtype, characterized by upregulated sugar and nucleotide metabolism. And then on the bottom here, MPS 3 in yellow, which is a mixed subtype, characterized by partial pathway dysregulation. If we look at this survival curve here on the right, we can see that in red on the bottom, the MPS 2 patients have significantly worse relapse-free survival compared to the other groups. So we can think of these tumors as kind of the worst of the worst. And so that suggests that the upregulation of sugar and nucleotide metabolism may be linked to highly aggressive phenotypes in these tumors. What Dr. Greer was waiting for, our lab focuses on the peroxisome proliferator activated receptor gamma, which is a mouthful, so we call it PPAR gamma for short. And this is a ligand activated transcription factor belonging to the nuclear receptor superfamily. And it's expressed in mammary gland epithelial and associated stromal cells. Endogenous ligands, such as fatty acid metabolites and thiazolidine diones, such as rosiglitazone, will bind, causing a conformational change, which results in increased transcription of target genes involved in adipocyte differentiation, as well as lipid and sugar metabolism. And previous work in our lab has shown that it also plays a key role in suppressing breast tumor progression. This is further supported by epidemiological studies showing that increased expression of PPAR gamma is correlated with better clinical outcomes, such as increased overall survival and decreased recurrence in aggressive breast cancer subtypes. There's also improved outcomes among breast cancer patients who were also taking who also had diabetes and were taking anti-diabetic PPAR gamma activating drugs compared to non-users. So there's not been a lot of work done to investigate the specific correlation between PPAR gamma and TNBC clinical outcomes. So given what we know about the role of PPAR gamma in regulating normal sugar and lipid metabolism, as well as its impact on breast cancer outcomes, further exploring the role it plays in metabolically dysregulated TNBC tumors and investigating the effect of its activation on key cancer-related pathways has the potential to unveil important prognostic information and improve patient outcomes. So a previous student in our lab named Liz began investigating the relationship between PPAR gamma and TNBC using an in vivo xenograft model. So for those of you who are unfamiliar with what a xenograft model is, essentially Liz took a representative TNBC cell line and she injected it orthotopically into the mammary fat pad of immunocompromised mice. So we can monitor tumor growth over time. So what you're seeing here is luminescent imaging of the parental cell line. So there's no PPAR gamma expression or activation here at weeks 1 and week 6. So we see a drastic increase in luminescence, suggesting increase in tumor growth. These mice are also treated with rosiglitazone, a PPAR gamma ligand. And qualitatively, we see a decrease in luminescence. There are also PPAR gamma wild type expressing cells. And again, we see another shift towards decreased luminescence. And finally, these PPAR gamma wild type cells, PPAR gamma wild type tumors were treated with rosiglitazone. And we see the most drastic difference in luminescence at the bottom there. You can also represent this graphically where we see by measuring tumor volumes that there is a significant decrease in primary tumor volumes with the expression and activation of PPAR gamma. And so with this, we wanted to take these tumors and metabolically profile them to assess PPAR gamma dependent and independent effects on tumor metabolism. So the way that I'm proposing to metabolically profile these tumors is using desorption electrospray, ionization, mass spectrometry imaging, another long word, so we call it DESI for short. And this is a non-destructive mass spectrometry imaging technique that allows for the detection of small molecule metabolites and lipids in tissue samples at ambient temperature. So essentially what's going on here, if you can see my cursor, is that high velocity ionized solvent droplets desorb the analytes directly from the sample surface here into gaseous droplets that then get delivered to the mass spec inlet. And that's where the mass to charge ratios are analyzed. Something that's important to note is that we conduct DESI at a resolution of 100 by 100 microns, and that is representative of 1 pixel. And each pixel has its own respective spectrum. DESI also retains the spatial resolution of its data, which is really exciting because it produces these 2D heat maps. And then we are able to overlay that with pathologically annotated tissue slides. And this is the same slide that will have been run on the DESI. And so this allows me to compare the two. And essentially what this means is that I can tell you where a metabolite is or where an ion is present on any given part of the slide. I just wanted to quickly go over the sample analysis pipeline, just so you can get a better understanding of some of the data that I'm going to be showing you later. So first, the tumor samples will be analyzed by a pathologist, or annotated, sorry, by a pathologist. So this is an example here. In our case, we're looking at tumor, necrotic, and non-tumor tissues. I then take the pathology annotated image and I overlay it with the DESI-derived heat map. So this is what you're looking at in this third picture here, is the overlay of the two images. I then use a software developed by one of our collaborators to plot what's called regions of interest, or ROIs, and these correspond to each of the pathological zones. So I'll plot regions of interest in the tumor region, in the necrotic region, and in the non-tumor region. So if you remember on the last slide, I mentioned that we DESI in 100 by 100 micron pixels, and each of these pixels has its own spectrum. So when we plot an ROI, that is the merging of 9 of the pixels together, and that forms one ROI spectra. The data we pull out from here is then normalized to the total ion current, and then I bin and filter the data, and then perform some univariate and multivariate analysis. So the DESI was used to analyze the tumors from the xenograft study that Liz did, that I mentioned earlier. And we applied a principal component and linear discriminant analysis, or PCA-LDA, to generate the plot that you're seeing here. So each dot on this plot represents an average of 9 of the pixels that I discussed on the previous slide. And there are quite a few, there are 2 main important things that I want you to take away from this figure. So the first is that the samples are clustered based on tissue type. So if you look over here to the left of the Z-axis, we're going to see tumor ROIs plotted on this side. And then on the right, or towards the bottom over here, these are the non-tumor ROIs. So the DESI was able to separate and accurately predict tumor versus non-tumor tissues. The other thing that I want you to pay attention to is that they're also clustering based on treatment group. So again, if we look, oh, there's my cursor. If we look over here, all the way on the left, these are the parental tumors. And then in the middle here, we've got the parental treated with Rosie, as well as the PPAR gamma wild type tumors. And then all the way over here to the far right / the top right, we have the PPAR gamma wild type Rosie treated tumors. So this is really exciting because not only can we distinguish between tumor and non-tumor, but we can also identify the different metabolites that are present between the different groups. One of the ions that was found to be present in these tumors was oleic acid. And this is a monounsaturated fatty acid that has been shown to promote the growth of highly metastatic breast tumors. So you can see here that the expression and activation of PPAR gamma significantly decreased the presence of oleic acid in these tumors. So we're looking at the comparison between red and green over here on the far right. So coming back to what we know about metabolism, this is an example of PPAR gamma signaling potentially shutting down a fatty acid source that might be fueling metastasis. So based on current literature, everything I just talked about and work in our lab, I hypothesize that the activation of PPAR gamma signaling, reduces the growth and spread of TNBC through changes in metabolic signaling. So to assess this hypothesis, my first aim is to define metabolic changes occurring in vivo preclinical TNBC xenograft models following activation of PPAR gamma signaling. And next, I want to characterize the in vitro phenotypic effects of activating PPAR gamma signaling in metabolically distinct TNBC subtypes using representative cell lines. So before we dive into the methods and the results, I'm just going to briefly talk about the human-derived TNBC cell lines that I've been working with. So if you look at this table, there are some key characteristics of the BT549 cell line, as well as the MDA-MB231 cell line, which I will refer to as 549s and 231s. What's important to note is that both of these cell lines endogenously express very low levels of PPAR gamma. I also want to take you back to this graph that I mentioned about the molecular pathway-based subtypes. So the BT549s were found to be part of the MPS2 subtype, or the glycolytic subtype, which is the subtype that I mentioned was kind of the worst of the worst with dysregulated sugars. And then the 231s are part of the MPS3, or the mixed subtype. All right, getting into the good stuff. So we can dive into the first aim, which is defining metabolic changes occurring in vivo preclinical xenograft models. So what we did was took these cell lines and then transduced them with a WPI luciferase and DOX-inducible PPAR gamma wild type vector. Then injected these cells into the fat pad of immunocompromised mice. So we have 4 different groups here. The first is the parental cells with normal chow. Then we've got the parental cells that are supplemented with a rosy chow diet, which is, as you remember, the PPAR gamma ligand. The third group would be the PPAR gamma wild type with normal chow. And then at the end here, we have the treatment group, which is the PPAR gamma wild type supplemented with the rosy chow diet. We can then follow these mice for up to 6 weeks using luminescent monitoring due to the luciferase gene that we transduced. And then at endpoint, the mice are necropsied, the organs are collected, and then we freeze them. These frozen tissues can then be sectioned and run on the DESI. And then that exact same slide that is run on the DESI then gets sent for H&E staining and pathological annotation. And then from there, I can conduct data and pathway analysis and correlate the effects. So similar to what I showed you earlier with Liz's xenograph study, I wanted to increase the N of that study. So she started with 3 mice per group, and now we have a total of 10 mice for each treatment group for a total of 40 mice. So again, we can see here qualitatively that there are differences in tumor growth from week 1 to week 6 in the respective groups. And we can see at week 6 in the parental group, the tumors are much larger. And then as we treat with rosy and activate PPAR gamma, the tumors get smaller. And we can also look at this quantitatively. So here you have a plot of average area by average density of the luminescence. So going from top to bottom, in pink here, we have the PPAR gamma, the parental tumors. And then in blue, after that is parental treated with rosy. The green is the PPAR gamma wild type. And then all the way on the bottom here, we have the PPAR gamma wild type treated with rosy. So this kind of reflects what we were seeing in the luminescence images in the middle there. And we're seeing that decrease in luminescence as we express and activate gamma. At endpoint, I then measure tumor volumes. And looking at this graph here, we can see that there is a significant decrease in tumor volume when we express PPAR gamma. So if we look at the difference between the pink bar here and the green bar. This is just with PPAR gamma expression. There's a significant decrease in primary tumor volume. And then if we look at the comparison between the parental and the PPAR gamma wild type treated with Rosie, we see an even bigger significant decrease in primary tumor volumes. I then took all of these tumors, so all 40, and ran them on the DESI. So I followed the same analysis framework that I went over earlier, and I was able to train a model and generate this PCA LDA that you see on the right over there. So this is a lot to look at, so we'll walk through it kind of slowly. So just as a reminder, I have 4 groups and 3 pathological zones. So we're looking at tumor, non-tumor, and necrotic. So we'll start with the parentals. The parentals are up here, and we're looking at tumor in green, non-tumor in yellow, and necrotic in orange over here. And then moving slightly in front, we have the parental treated with Rosie. So we've got tumor in dark blue, slightly lighter blue is non-tumor, and then the necrotic is hard to see, but slightly behind right here. Then in the bottom left corner, we've got the PPAR gamma wild type samples. Ooh, so sorry. And so we've got tumor again, non-tumor in this like cyan color, and then necrotic. And then all the way at the top over here, we have the PPAR gamma wild type treated with Rosie. So we've got the tumor, non-tumor, and necrotic just behind. So what's interesting to notice is that not only is it separated by the groups, but something that I thought was kind of interesting is that the tumor and the necrotic ROIs clustered more closely together than the non-tumor ROIs, which is to be expected, but it's also exciting that we're seeing that reflected in the analysis. So out of the 15,243 ROIs that were plotted, 15,061 of them were correctly predicted, while 182 were incorrectly predicted. So that gives us a balance accuracy of 97.56%. So this essentially means that this model is able to predict ROIs belonging to 3 different pathological zones within 4 treatment groups with a balanced accuracy of 97.56%, which I thought was very exciting. And then I wanted to kind of take this further into the analysis. So I started by taking a look at ions significantly elevated in the parental group versus the PPAR gamma wild type group treated with Rosie. So essentially, what I wanted to do was look at the effects of PPAR gamma expression and activation compared to controls. So to do this, I first ran a t-test to identify ions that were significantly elevated in the parental tumor regions, and then sorted these by p-value, pulling out the top 25 most significantly elevated ions. I would love to go through all of them, but we don't have time to go through 25 ions. So this is just a shortened list of some of the ions that popped up in the top 25. Very interestingly, we see gondoic acid and its C13 isomer here. So we see some bar graphs. So in the dark pink, we have the parental tumors. Light pink is parental non-tumor region. And then we have the PPAR gamma wild type with Rosie tumor region. And in the light blue is the PPAR gamma wild type with Rosie non-tumor. So this is interesting because recently published work in our lab actually showed that elevated levels of gondoic acid are associated with lymphovascular invasion in colorectal cancer. And this is a prognostic feature that's associated with poor outcomes in this cancer type. And so this significant difference in gondoic acid in these xenograft tumors could reflect a more aggressive phenotype in the untreated tumors here. Also within the top 25, I noted 3 other long and very long chain fatty acids to be elevated in the parental tumor regions compared to the Rosie treated PPAR gamma wild type tumor regions. And so these 3, arachidic, uric, and cirrhotic acid, are all saturated and monounsaturated fatty acids that are involved in like elongation and synthesis. Sorry, they're intermediates of saturated and monounsaturated fatty acid elongation and synthesis, and that contributes to cancer cell proliferation. So again, if we're looking at the bar graphs along the bottom here, we can see that there's a significant difference in ion intensity shown between the parental tumor regions and the treated PPAR gamma wild type tumor regions. And we observe this trend across the board here through all of the different ions that I've identified on this slide. And we also observe this trend when comparing non-tumor regions, which I mentioned were the light pink and the light blue here. And so this kind of highlights the systemic effect of PPAR gamma expression and activation in these samples. We can also visualize these ions that I just talked about in relation to the pathologically annotated slides. So on the left here, we have 4 panels, and this is the parental representative images. And then on the right here, we have representative images of the PPAR gamma wild type rosy treated group. And so we see here in red, that's the tumor region. Orange is necrosis, and then the highlighted is the non-tumor regions. And so what I think is really cool is when we look at these parental images for the ions, we see this like pretty stark contrast between the tumor region and the non-tumor regions when it comes to ion intensity. Whereas when we look at the PPAR gamma wild type and rosy treated group, we bring the images to make them more visible on screen, but there's less of a contrast between the tumor and the non-tumor regions here. And we see that for this ion as well, gondoic acid, this one down here, as well as this ion on the top right over here. So essentially what we're seeing here is that there is a greater ion intensity of these ions in the tumor regions, whereas in the parental group, whereas in the treated PPAR gamma wild type group, it's more of a spread out along the tissue. So why does this matter? I don't know if you remember when I earlier mentioned that fatty acid synthesis and oxidation are important in cancer cell metabolism. And a number of the ions that I identified here are involved, as I mentioned, in the elongation and, the elongation of saturated acid to cirrhotic acid and the elongation of oleic acid to nervonic acid. And this is really important because de novo fatty acid synthesis occurs in tumor tissue, and this leads to increased concentrations of acids that are further converted into saturated and monounsaturated fatty acids. And then these can then act as building blocks for much larger, more complex glycerophospholipids, which then get used to form like cell membranes of rapidly proliferating cancer cells. In terms of ongoing studies for my first aim, I want to continue to probe into this DESI data that I have and look at other treatment groups and expand. I also hope to conduct the xenograft study using the other cell line, the BT549s, and conduct similar analysis, and then assess the differences in metabolite abundance between these two models. Bringing it back to the cell lines of interest, we're going to move into my second aim, which focuses on in vitro work. So if you'll remember, I mentioned that the BT549 cells are more of that glycolytic subtype, while as the MPS3 are the mixed the 231s are the mixed subtype. And so we were wondering what would happen to these cells if we altered the glucose levels and activated PPAR gamma. So essentially what we're hoping to do here is mimic conditions similar to what you would see in an individual with type 2 diabetes to assess the effects of sugar concentration and PPAR gamma expression and activation on the invasive potential of these cells. So I conducted a Boyden chamber invasion assay, and I introduced the parental and the PPAR gamma wild type cells to different glycemic conditions. So we did normal glycemic and hyperglycemic conditions, and these concentrations reflect physiological glucose concentrations. These cells then get seated on top of a matrigel layer in a 96-well plate, and then they get to invade through the matrigel to a filter on the bottom towards a chemoattractant. Then we fix and stain the filters with DAPI, and then we can visualize and analyze them. So I don't know if you'll be able to see. Where's the light? That looks really cool. Okay, so here we have some representative images of the DAPI stained filter from the hyperglycemic condition. So we can see here that when we have the parental cells, there's a lot of blue, a lot of invaded cells. And then when PPAR gamma, this should say wild type, I'm very sorry. When PPAR gamma is expressed and activated, we see that there is like a decrease in the number of invaded cells. Again, we can look at this quantitatively. And so if we look at this bar graph here, the first thing that you'll probably notice is that the cells are much happier to invade under hyperglycemic conditions. So when they have more sugar, more cells are invading through, they're a lot happier. And we do notice similar trends between the normal glycemic and hyperglycemic condition. However, the statistical significance lies in this hyperglycemic condition, where the expression and activation of PPAR gamma significantly decreases the cell invasion. Turn the lights back on. So even though these cells have dysregulated glycolytic pathways, it's unclear whether they may also be impacted by changes in lipid metabolites. It's important to note that patients with type 2 diabetes, there's also often dysregulated lipidemia, oxidized lipids, and this can play a role in promoting tumorigenesis. So we wanted to see if we could replicate this in vitro and assess the impacts on migration potential. So these cells were seeded into a 12-well plate, and then we scratched the plate here. So these are little scratches. The light pink is representative of the parental cells, and then the dark pink are the PPAR gamma wild type cells. I then treated with a combination of rosiglitazone, oxidative low-density lipoproteins, as well as a PPAR gamma inhibitor. And this will allow us to assess the PPAR gamma dependent versus independent effects, which is just what you see described here. I then assessed these scratches and photographed them at 3, 6, 9, and 24 hours. And then this is kind of what you see here on the left. So we have representative images of the PPAR gamma wild type cells with the various treatments displaying wound closure. So at 0 hours, the wound is fresh, it's just been scratched. And then we can see as we progress over time to 9 hours, premature, the cells start to invade into the wound and close that gap. So again, looking at this graphically, this graph can be a little confusing, so I'm going to walk you through it. On the bottom here, we have the first 4 bars, which are the parental cells. And then the next 4 bars would be the PPAR gamma wild type cells. And these are treated sequentially with vehicle control, the oxidated lipids, rosiglitazone, and then a combination of the two. So that would represent these 4 as well as these. And then at the end over here is when the PPAR gamma inhibitor was introduced. So it's not currently significant, but we do notice a slight increase in migration in both the parental and the PPAR gamma wild type cells when we introduce the oxidative lipids. We can see that here in blue. And then it slightly trends downwards when we introduce rosiglitazone. Again, this is only an N of 2, so it does need to be repeated, but it's just an interesting trend to note. What's even more interesting, though, is at the far right over here, looking at the purple bars as well as the yellow and maroon. We can see that cells that have inhibited PPAR gamma and have been introduced to OXLDL show significant increases in cell migration compared to the vehicle controls of both the parental and the PPAR gamma wild type cells, as well as to the parental oxidative lipids cells. We also see a significant decrease in migration when we treat these inhibited cells exposed to oxidative lipids with rosiglitazone. So I hope to continue doing these migrations and get one more done. And I would also like to conduct some invasion and proliferation assays with the previously described treatment groups and add in some dysregulated sugars. I'd also like to assess protein and RNA using Western blotting and QRT respectively, and then correlate my findings here with my in vivo studies. So that was a lot of talking. So essentially the main takeaways are that DESI signatures can differentiate between tumor and non-tumor regions, as well as the treatment groups. The ions involved in saturated and monounsaturated fatty acid elongation are elevated in parental tumors versus rosy-treated PPAR gamma wild-type tumors. PPAR gamma expression and activation significantly decreases BT549 cell invasive potential under hyperglycemic conditions. And the introduction of oxidized lipids paired with the inhibition of PPAR gamma significantly increases BT549 cell migration, which then is significantly decreased when treated with rosiglitazone. In terms of future directions, I hope to explore, or not I, just future in general, explore the metabolomic profiles of human TNBC samples and correlate the in vivo and in vitro metabolic targets with human TNBC clinico-pathological information. And in terms of long-term significance, this hopes to reveal metabolic pathways that could potentially be targeted using therapeutics, inform clinical decisions, and provide prognostic information and improve quality of care, patient outcomes, and overall survival. With that, I would like to acknowledge my lab members, my collaborators, Chris, my supervisor, my thesis advisory committee, and Dr. Greer, of course, and all my funding support. Thank you all for listening.
Ready for some questions now?
Do I have to do that part?
Yeah. Oh, we have a question.
Why? Yeah. Thank you.
I was just curious about how you determine your endpoints for your in vivo, because you guys kept mice in study for 6 weeks.
Yeah.
And I just, because you're going to do a treatment, there was a specific reason for why for the 6 weeks.
Yeah, so we carry them out. Typically, like with the parental cell line, we inject 1 million cells. And so these form tumors quite quickly, and they grow and metastasize. And when they metastasize to the lungs or the brain, that would be a humane endpoint. So at 6 weeks, typically is when the tumors reach size. And to be comparable across groups, we take them all out to 6 weeks to maintain the consistency. But yeah.
Okay.
For your presentation,
Thanks.
did you guys look at metastasis colonies, like at your endpoint? Or do you have evidence of metastasis?
Yes, so we do, we
try to repeat the question.
oh, sorry. She was just asking if we look at metastasis to the lungs, or other sites. The answer is yes, we can look at that. And so we can assess that using like the imaging software, with GFP, we can look for the presence of tumor cells in these, mice. And we can also take these, tissues and run them on the DESI as well to assess the Mets. I haven't done that yet, but that is something that would be really exciting to do.
So you mentioned that, I guess it was oleic acid that you say promotes metastasis, or is it tumorigenesis in general? Is that understood mechanistically, how that might work?
That's a great question. I did say that. Yes, I have read, there are papers that suggest that it does promote the metastasis of breast tumors. I can't say I'm 100% familiar with like the mechanism behind that,
Maybe it's not known.
but yeah, I'm not entirely sure, but
well,
definitely something to think
I mean,
about.
that's sort of, I think, a big question that arises from your work. So you're seeing interesting differences in metabolites,
Yeah.
and very often, I guess it's selected by the technology which things you can observe with DESI, but how many of these you can actually point at literature that supports mechanistic involvement. I think are any of them in pathways? Are they produced in pathways that there's a known role for PPAR gamma in regulating, for example?
For the ones that I mentioned specifically, I haven't found like a direct link. I haven't consumed all the literature possible, but there are ions that we do pull up that are directly related to PPAR gamma, and we can do more targeted analysis of those metabolites using like more destructive mass spec techniques down the line. But pathway like analysis or mechanistic pathway identification is something that definitely requires more study.
This is one thing I can think of as an example is we must know by now what are the target genes that PPAR gamma can regulate. And so are any of those encoding enzymes that are in these pathways that are involved in regulating metabolism? Fatty acid metabolism in particular, I guess.
Are you saying like, did I pull any of those up like in my?
Well, when you're profiling
Yeah.
the analytes that you can identify with DESI,
Mhm.
do any of them map into metabolic pathways where there are enzymes involved in those pathways that are targeted by PPAR gamma?
Most definitely, yeah. I haven't like with the DESI data, we pull up like the top 2,000 ions. And so currently this is like very like preliminary analysis of my own work. But I'm pretty positive that we'll be able to pull up some things that are related to PPAR gamma for sure.
Other questions?
Yeah.
I'm just wondering, I don't think you explained it or not, but how do you select like the RLIs? How do you select the specific regions that you're going to be looking at? So you kind of
Yeah.
put random dots there.
Yeah, that was just kind of like to show representationally, but essentially in the software that we use. Let's see if I can, okay, I'm gonna do it on this one, but we will do it here. But so essentially what you do is you effectively color in this region. So on the software, you can like plot the ROIs and you cover the entire region with one specific, so per pathological region, you like plot differently. And then we can like amalgamate that for all the samples across like all 40 samples, like we bin and filter them all together. And the software itself will merge the pixels and average the spectra. So it's not like scattered like this, like they're side by side. Yeah, this is just kind of to show like visually, but yeah. Does that make sense?
Yeah.
Okay. Yeah.
You may have mentioned this and I might have missed it, but you mentioned in colorectal cancer you've also done this sort of analysis. Do you see similarities in the types of things that you're detecting across different cancers, and like how would this apply to other subtypes of breast cancer as well?
Other subtypes of breast cancer, I'm not sure, but a lot of the ions that I presented today, shameless plug for Natasha's paper and Martin's paper, you should go read it. But a lot of the ions are similar that we pulled up. So all of those like fatty acids that I mentioned, the long chain and very long chain, were also pulled up in the colorectal cancer paper. Yeah.
Yeah, I had a great presentation.
Thank
I
you.
have a couple of questions. One is a really fundamental question about your background.
Yeah.
I've sat through many presentations on breast cancer, but I didn't know enough about estrogen receptors to be able to access this. So are these missing all estrogen receptors? I mean, there are different types. So when you say ER negative, what do you mean exactly?
Like as in like it doesn't express that estrogen receptor? Am I
Well,
misunderstanding?
there's more than one.
Oh, I see.
So do we know?
That's good.
Yeah, when people say ER negative, what do they mean in terms of this?
Yeah, that's a good question. I'm trying to think of a way to phrase it that makes. Actually, that's a great question. I'm not sure. Like, I don't think it would be missing like all the different types of estrogen. I want to say it's alpha, but I don't know.
And to my knowledge, there's alpha, beta, and there's a G coupled protein estrogen receptor.
Yeah.
So are they, when you say ER negative in TM triple negative,
Yeah.
is it always the same estrogen receptor that's missing, or are they all missing, or does anyone know? People must know.
Yeah, I
Yeah.
don't know.
Bet you Chris knows.
Is there a pathologist in the room? Sonal, are you there? Can you hear us?
Yeah. No.
When you do IHC on breast cancer, what do your antibodies see?
Yeah, I'm not 100% sure.
I would imagine it's a cocktail of at least alpha and beta.
Yeah.
I don't know.
Something to find. People must know. I believe it's
Oh, yeah.
should help. So you showed some nice data where, with your DESI, you can now correlate that to regular, histology. Right?
Yeah.
Yeah. So spatial omics is kind of the real thing these days. Everyone's doing this stuff. So what else can you do with DESI? Can you do multiplex immunofluorescence? Can you do spatial transcriptomics? You know, other things you could overlay on what's happening with the metabolomic analysis you're doing.
Yeah, so for right now, like because it that's a great question. I was actually talking about this earlier today, the spatial transcriptomics. But for right now, I don't think you can do like the spatial transcriptomics or IF after you've H&E stained that direct slide. So for right now, when we are analyzing our DESI data, like this slide is like fixed and stained, but you could take this like DESI data slide and then do other like spatial techniques on the slide that comes out.
Tells that wanted to do multiplex immunofluorescence.
Yeah,
You could.
yep. No, I would think so.
He'll be coming to visit you to ask you how to do
The DESI part, sure. The multiplex, not familiar.
Another PPAR gamma question. So PPAR gamma, you showed in your introduction these interesting observations or correlations between people taking Rosie, or I forget which cortisone they're taking for diabetes, but one of the cortisone drugs that are ligands for PPAR gamma. So that's very intriguing, and we guess we've known about this for how long, Chris? 10 years plus? These epi data. So it's very intriguing and supportive. Is there other kind of genetic like GWAS studies that would implicate either loss of or gain of function type mutations in PPAR gamma that correlate with differential prognosis?
Yeah, I believe.
In any cancer for that matter.
I have read a few studies that talk about like PPAR gamma gain and loss of function, and I've tried to do some analysis myself using the TCGA data, but PPAR gamma expression is relatively underreported in the breast cancer cohort in the TCGA. But I have read a few studies. There was one that I don't know the name of the author that talked about I believe it was a gain of function mutation in PPAR gamma.
That'd be something to-
Yeah, I have it in my folder. I can't recall the cancer type that it was looking at though.
Sometimes
There are some point mutations that increase risk in colorectal cancer.
in this sense, right?
In this sense, but there's actually very few. There's more changes in localization of the transcription factor. It doesn't have this, it's not a P53 mutational hotspot, but it's more presence and ability to function.
Yeah, it's just some of these, like the UK cohort, it's so huge, there's over 400,000. Sometimes you can tease out very, very small effects. And they're not necessarily missense mutations, they can be SNPs that are associated with slight changes in transcription even. But because it's so powered, you can sometimes see. Because I'm getting the impression that this is what PPAR gamma is. It's not a massive effect type immunosuppressor, but it's got some impact. Yeah. Okay. I don't see any questions on the Zoom. If you're there and you have a question, you can use the raise hand function or drop it in the chat. Any other questions from the audience? Last chance. Oh, she'll be back.
You could send me an email.
Okay, then join me in thanking Madeleine for very interesting.